At this WIN Symposium, I'm presenting the data of RET inhibition in RET aberrant cancers. So RET is called the receptor tyrosine kinase. It's rearranged during transfection. It's an oncogenic driver seen in multiple cancers. It's seen most commonly in lung cancer, which is 1 to 2% of all lung cancers. And then RET fusions are seen across multiple cancer types, less than 1%. In papillary thyroid cancer, RET fusions are seen in 10 to 20% of papillary thyroid cancer. And RET mutations are seen in 100% of hereditary thyroid cancer. And then in over 60% of sporadic medullary thyroid cancer. And we see RET fusions and aberrations across multiple other cancers. And we have a great interest in RET starting from 2017 because we have highly potent selective RET inhibitors that can have a great benefit for patients with RET activating aberrations. And my main presentation was on the preclinical and clinical data of the selective RET inhibitors. The first one was the BLU-667, which is a highly potent selective RET inhibitor. And based on preclinical data, it was selective against the RET across multiple preclinical models. And it also addressed the gatekeeper aberration. And it was off target, it was much better, and it was more selective for RET than the multi-kinase inhibitors. And the early phase trial was promising. Based on that, the expansion cohort was established. And the recent data was presented at ASCO, and I presented the updated data in the WIN Symposium on those updates. So based on the early phase data and the expanded access program and the expansion cohorts, now BLU-667 has 2 US breakthrough designations. One is for RET fusion positive thyroid cancer, and the second one is for RET mutant medullary thyroid cancer. The second drug I spoke about is the LOXO-292, which again is a highly potent and selective RET inhibitor. A preclinical model showed that this was active against multiple histologies compared to TKIs such as vandatinib and cabozantinib. It was also shown to have activity in orthotopic brain tumor models. So based on this, we initially treated a patient as a single patient compassionate access IND with this selective targeted therapy. And this patient had progressed from 7 multi-kinase inhibitors and had developed a secondary gatekeeper aberration and had a fantastic response to the LOXO-292, which is a selective drug. At the same time, the first in human phase 1 study was started, and now we reached an recommended phase 2 dose, and then expansion cohorts are ongoing, and the response rates are fantastic for a phase 1 and expansion cohort. So LOXO-292 is very active in RET fusion positive lung cancer, RET fusion positive pancreatic cancer, RET fusion positive papillary thyroid cancer, and RET mutant medullary thyroid cancer. The most common RET mutation that we see in medullary thyroid cancer is RET M918T. It is active against that. And we also see secondary gatekeeper aberration in RET mutant patients that have been treated with vandatinib or cabozantinib, and some patients who have germline aberrations. This drug is pretty active. And it's rewarding to see patients derive clinical benefit and have dramatic responses from these selective RET inhibitor therapies. And based on the early phase data and the expansion cohorts, LOXO has received 3 US breakthrough designations. One is for RET fusion positive non-small cell lung cancer. Second one is for RET mutant medullary thyroid cancer. And the third one is for RET fusion positive papillary thyroid cancer. So these drugs are on fast track and have received breakthrough designation. An exciting times for RET aberrant patients. The main thing is that we need to start with genomically testing these patients. So one might play to patients and physicians is to undergo genomic testing, and if you have specific aberrations like RET, please get enrolled in one of these selective RET inhibitor studies. And again, that's true for all genomically driven cancers. As you know, recently we have NTRK inhibitors being approved for NTRK fusion positive cancers. So it's possible in the future that we can do a broad-based genomic testing and identify patients with specific genomically driven cancers and match them to targeted therapies so that they can have the clinical benefit from these therapies. Another thing is that most of these patients are being treated at the last minute, last round when they're relapsed and refractory from multiple other therapies. Since these drugs are highly potent and active, I think it would be reasonable to take it to the first line setting when we can see very good responses with these drugs. I'm looking forward to taking these drugs to the front line setting.